DNA structure and transcription. So I just was talking about the ribose sugar that's going to be a part of DNA. There's the phosphate. That phosphate will attach right here. And then we'll have another ribose that attaches that phosphate, then another phosphate, and it's going to alternate between ribose and phosphate to form kind of the chain of DNA. And this shows the bases, the nitrogenous bases that are part of that. And so the nitrogenous base is going to link on the inside right here. So the phosphate and sugar form what we call the backbone, and then the bases will stick out into the middle. And the bases will actually form a bridge across to a second strand, a second backbone of DNA that's going the other direction. And so, and it will attach one base to another. A's will match with T's and G's will match with C's across that bridge to form the DNA structure. And we can see that kind of depicted here. We have the backbones made of sugars that are red, and then the little purple dots are phosphates, and they go through and form, kind of twist around each other in that double helix shape. Then we have the bases in between. And the bases, A's with T's, G's with C's, to kind of link those backbones together. DNA is not the only nucleic acid. There's also RNA, which very similar, but there's some key differences. So one of the key differences is DNA is double-stranded, RNA is single-stranded. DNA uses A, T, G, and C. RNA uses A, G, and C. Instead of T, it will use uracil or U right here. And if you look chemically, they're very similar, right? The difference is primarily that thiamine has a carbon with hydrogens coming off of here, whereas uracil just has the hydrogen there. But they'll behave very similar chemically. The U will match with an A, just like the T matches with an A. And the last difference is pretty slight, but if you look right here, DNA, deoxy, meaning it's missing an oxygen. It's its name because in this position right here, it has a hydrogen, and in this position, it has an oxygen and a hydrogen. So it's missing an oxygen. That's why it gets the name deoxyribose. So those are the three key differences between DNA and RNA. They also serve different jobs that they're able to do because they have those different functions or those different structures. And as we go through that, hopefully it will become apparent. So when we relate this now to genes, the segment of DNA, right, we are going to actually have two strands of DNA there. So which one then becomes the code? Well, we have the one that carries the code that makes up your gene. We're going to call that the coding strand. So in this case, let's say it's the top right here, right? That would be the coding strand. The strand that pairs to that, we call the template strand. What you need to do, and I know in the lesson it kind of went through and talked about how the coding strand works and template strand works in this process that we're going to talk about called transcription. What we want to understand is that the coding strand is what has the code, right? And what our cells do is it takes the coding strand and it builds an RNA copy of it. And the reason why is because we want to keep the DNA safe inside the nucleus, and we want to send messengers out to carry the instructions. If we don't keep the DNA safe, that DNA could become damaged, and that damage will be passed on if we do that. So it's kind of like if you want to use your grandmother's recipe book, you don't want to take that recipe book and throw it out on the counter right where you're putting all the ingredients together. Instead, what's common is to go and actually write down the recipe out of the recipe book and then have that card with the instructions to act where you're actually going to put together the item that you're baking, right? And so the DNA is going to be the recipe book. The RNA is going to be the card that you wrote the instructions down on. It's going to be what goes out of the cell. Now, there's more to it than that, but hopefully that analogy helps you understand the relationship that they have to each other. So we have here a strand of DNA, right? And it's hard to fit it all in one place. So we just got to imagine this strand is continuing on down through here until it reaches the end. Now there's some terms or some notation here I want you to notice. There's 5' and 3'. That's referring to which part of the ribose is there. So when we name the carbons on the ribose, we start from the oxygen position and we call the first carbon after that, that's the 1', 2', 3', 4', and then this one up here would be 5'. So when we talk about the prime, we're saying that the way that this is oriented is the 5' is up. So this 5th carbon that's right here is oriented up. If instead it was 3', this whole image would be flipped upside down. This bridge that's formed between our DNA molecules, let's see, the bridge that's formed, if this one is 5', if that 5th carbon is to the left here, the opposite side will be 3'. So it will be flipped the other way. So if this one is oriented like this, that top strand there, the bottom strand will be oriented like this. And that's what we call anti-parallel. So this DNA is anti-parallel to each other. And that's what this is showing us, right? The 5' end is here, and it runs until it reaches its 3' end, and the other strand goes the opposite way. Genes are read 5' to 3'. So we're going to look if our DNA gene is the hemoglobin gene here, we're going to read it going from left to right. If we were using the myosin gene in contrast, it goes from right to left, right? Because it's on the bottom strand now, it's got to go 5' to 3'. I'll always make it clear what you're being asked to do. And typically I'll just give you one strand of the DNA. I won't give you both strands. And so we'd start here. We start at the TATA box, which it mentions in the text here. That's going to be the promoter. That's where we will start making our RNA copy. And we will go all the way through this letter by letter until we reach the terminator. So you're going to take and make an exact copy of what's in the coding strand, except you're going to replace T's with U's. So our coding strand, our RNA that we make from this coding strand would be U, A, U, A, and so on until we reach the terminator. That is transcription. That process is done by an enzyme. Again, enzymes help chemical reactions happen, called RNA polymerase. So RNA polymerase builds an RNA copy of the DNA code, and that is transcription. That process includes more than the instructions. The instructions are actually the bolded parts right here. That's including stuff that we don't need. Before we can go on in this process of gene expression, we have to modify or process the RNA. So we just have the important parts. That's called RNA processing. These unbolded regions are what we call introns. Introns are not part of the message that we want to use. Exons are the bolded parts. That is the message that we want. In RNA processing, those regions will be cut out by something called the spliceosome. As those regions are cut out, we will then fuse the bolded regions back together to form just the exons of the DNA. So all this would be trimmed off. These sections in between would be trimmed off. Our DNA structure is getting much, much smaller as we process it. Or sorry, our RNA structure is getting much smaller as we process it. And then we'll just have those. Now there's an additional part to RNA processing. Once we get it down to just the exons, we will also put some protective parts to it. 5' cap and the poly A tail will be added to it just to help us get protection. They're not actually part of the message. They won't be read. They're just going to be that initial part. The protective part of the RNA. So that is DNA structure, and then transcription, and then RNA processing.